everyone. In this video, we are going to talk about the structure of DNA. Now, we know about DNA, right? It is, let's try to write the full form of DNA. It is deoxyribonucleic acid. So, deoxyribonucleic acid, DNA, right? So, the name is basically because of the fact it is deoxygenated, okay? At one position, if you try to see or look into the structure of the ribose sugar, we'll be soon talking about the components. We'll see there is a lack of oxygen at one particular position. So this is known as deoxyribonucleic acid. Now, the important thing is that the DNA, it is a genetic material. Genetic material, right? Genetic material of most of the organisms. Now, why do I say most? Because there are some organisms we can refer to not as organisms. Basically, there are some viruses which has RNA as the genetic material. So, basically, most of the organisms, the living organisms, they have DNA, deoxyribonucleic acids as the genetic material, and some of the viruses, they have RNA as their genetic material. Now, the question is, what do we mean by genetic material? Genetic material is the most important part or component of the cell which is present inside the nucleus. Why? Because whatever a cell is performing, whatever we are performing, everything is encoded inside that genetic material, which is the DNA. Okay, so whatever proteins are synthesized, whatever hormones are synthesized, whatever secretions are happening, how a particular cell is functioning, it is all encoded inside that DNA molecule or inside that genetic material. Now, you may be thinking, why DNA is the genetic material and not RNA? Yes, the important answer about this is DNA actually is a much more stable structure, specifically because of the fact that is deoxy, right? But in ribonucleic acid, that is RNA, they have that oxygen, and actually because of the presence of oxygen, it can easily be cleaved by various enzymes. So you understand why DNA is the genetic material and not RNA. So DNA is a genetic material. Now, this genetic material, as I have already mentioned, that it is mostly present in the nucleus, but there are also some genetic material, DNA, which is present in mitochondria. Also, we may find some DNA molecules in the chloroplast, right? So these are also the structures. So you must remember, questions are often asked that what are the places where we can find DNA other than a nucleus? So answer will be mitochondria and chloroplast. Now, the very interesting question we would like to discuss is how the DNA exists inside our cells. How DNA exists. Now, we should understand why are we asking this question. So let's talk about a great example. If we compare the cell structure and the size of the nucleus, if we compare that, then the size of the nucleus is very, very small as compared to the size of the cell. Now you see, the length of the DNA in human beings is about 2.2 meters. It is huge. So each DNA molecule, it is 2.2 meters, and you imagine that it is fitting inside that very small nucleus of the very small cell. So you understand how small or how importantly it has been coiled. Yes. So now we'll discuss how this length of 2.2 meters has been coiled, right? So if we just try to draw the normal structure of the DNA, it somewhat looks like this. It is double stranded. So we'll be talking about the structure properly. This is the double stranded DNA. Slowly and steadily it starts coiling. Okay, so it starts coiling like this. Okay, it will start coiling. This coiling is actually brought about by certain proteins which are present. These are the proteins which help in the coiling process. These proteins are known as histone. histone proteins. These are basic proteins, and these proteins actually help in coiling. Okay. Now, further, this is being coiled. This is being coiled. It is coiled very much, and slowly and steadily, what we get? We actually get the chromatin. Actually we get the chromosomes. It is very highly coiled structure, and what we have inside the cells, they are the chromosomes. Okay, you understand? So this actually is the part which contains that 2.2 meters of the DNA which is present inside the cells. Sugar. Okay, it contains the ribose sugar. The second thing which it contains is phosphoric acid. Phosphoric acid. And now the third thing is known as the nitrogenous bases. Nitrogenous bases. Okay. So it has got ribose sugar, it has got phosphoric acid and nitrogenous bases. Now let's try to draw the structure of the DNA which we were trying to draw here. Okay. If you open up the structure of DNA, it somewhat looks like this. Okay. So you see there are two strands. Okay. And very importantly, it is a, you should understand, you should remember this, that it is a double helix structure. Double helical structure. Okay, it is a double helical structure. Now, let's try to understand the components and the structure. Now, ribose sugar and phosphoric acid are these two areas. Okay, these strands, these are known as the backbone. It is basically called as the sugar phosphate backbone. So these lines, these strands you're seeing, these are sugar phosphate backbones. And now we have the nitrogenous bases over here. So first, before drawing the nitrogenous bases, we'll try to understand what are the two types of nitrogenous bases. First, we have purines and we have pyrimidines. Okay, purines and pyrimidines. These purines we are talking about, it actually has two nitrogenous bases. They are known as adenine and guanine. Okay, adenine and guanine. Whereas pyrimidines, these have cytosine, and it has thymine. Okay, so you understand there are four nitrogenous bases: adenine, guanine, which are purines. These are double ring structures, and cytosine, thymine, these are pyrimidines, which are single ring structure. Now they are present like this. Suppose guanine, here we have cytosine, and they are bonded by a triple bond. We'll be talking about the structure of the double helix in the next video. Now here just let us take a look how they are arranged. So here we can have AT, right? So like this, all these are bonded together, correct? And this structure is actually held together by these type of bondings. So here we have these bondings, and hence we have this double helical structure. So in this video we have talked about the structure of the DNA. We have seen the DNA, that is deoxyribonucleic acids. DNA is the genetic material. Mitochondria and chloroplasts are the additional structures other than nucleus in which we can find DNA molecules. We have talked about how the length of 2.2 meters DNA, how they are present. Okay, so it is highly coiled with the help of some proteins known as histone proteins, correct? DNA has a double helical structure. If you have seen a spiral staircase, right? If you have seen a spiral staircase or a spring, that is actually double helical structure. So you can imagine how a double helical structure looks like. It looks like somewhat like this. It has got three components: ribose sugar, phosphoric acid, and nitrogenous bases. Nitrogenous bases is of two types, purines which consist of adenines and guanine, and pyrimidines which consist of cytosine, thymine. Pyrimidines are single ring membered structures and purines they are double ring structures.